this is TS of monocot root. We will be studying in detail the transverse section of monocot root. Why we are studying TS of monocot root, or how we can differentiate monocot root from dicot root is, monocot root doesn't have secondary growth. So in this TS of section, the first and the foremost layer will be the root hairs. As we see here in the diagram, root hairs are arising from the pink color cells. Those are epidermal cells, and root hairs are those which are helpful for the absorption of water from the soil to the plant body. Next layer is the epidermis. Epidermal cells are the, it is similar to that, epidermis is almost similar to that of the dicot root. Here epidermis is single layer protective structure. Epidermis which are present in multiciliate condition in some epiphytes like ericaceae, those epidermis are not called as multiple epidermis, those are called as velamen. Velamen are nothing but these are a type of epidermis tissue which are helpful for the absorption, and it also gives the protection to the plant body. The next layer will be the cortex. Now what is the cortex? Here we have studied earlier that before cortex we can talk about a term called as exodermis, which is almost immediately next to the epidermis. They are generally present in one or more layers of cells, and this with reference to exodermis we have talked when we were talking in dicot root. Cortex is several layers of cells, made up of several layers of cells, and it consists of lot of intercellular spaces in between them. For example, if we take an example of maize, some layers of cortex below the epidermis are made up of thick walled cells, and these are often distinguished as exodermis, and these develop suberized secondary walls. But these suberized secondary walls differ from the Koch cells in possessing cell content in them. Next layer when we will be talking about is endodermis. Here endodermis we can call it as an innermost layer of the cortex, and is characterized by the presence of Casparian strips in them on both the radial and transverse walls. In monocots like in maize or in grasses, the radial and inner walls are extremely thick and as compared to the outer walls. Now, when we say the outer and inner walls thickenings are this, these thickenings of the inner walls of the endodermal cells is due to the development of a type of lamellae of suberin, and at the latter stages of development, this suberin lamellae is covered by a layer of cellulose, which in sometimes in monocot roots attains a considerable amount of thickness. Okay. In addition to this, these from here and there, some of the endodermal cells remains thin walled, and these cells are called as passage cells. What is the use of passage cells? We see the passage cells in endodermis, and these passage cells are useful for the radial diffusion of the water, and these passage cells remain thin walled throughout the life of the root, and in some cases they may also become thick walled, and it almost resembles similar to that of the endodermal cells in the dicot in all respect. Now next immediate layer to the endodermis we will be talking with reference to pericycle. Now what is pericycle here? In monocot root, the pericycle is usually uniseriate and is composed of single layer of thin walled cells. It may be normally it is uniseriate, but for example if we take an example of maize, you can see it as a multiseriate parenchymatous layer. And what is pericycle? Pericycle is generally nothing but a complete ring of cells. In monocot root you will see the presence of pericycle in a complete ring of cells, but in some grasses the ring is broken down due to the due to the breakage which is done because of the presence of some protoxylem elements. Now main function of pericycle is it give rise to the lateral roots. Okay. Next, when we come to the vascular tissue system, what do we have in vascular tissue system? Vascular tissue system, as you can see in the diagram, you have red color of xylem strips with that of the alternating phloem strips are present. So now here this is nothing but we can say vascular tissue system is nothing but an alternate composed of alternating strands of xylem and phloem. Now the number of strands varies in different plants. So in general we can say that in monocots the condition is polyarch. Why we are calling it as polyarch? Because the number of vascular bundle strands which are alternating are more than 4. Many numbers, so that's why we called it as a polyarch condition. Here we said it is a polyarch condition. In Palmae or in Pandanaceae family, you will see the number of xylem and phloem strands occurring more than 100 or even more number of xylem and phloem strands can be seen. So that's why we call this condition as polyarch conditions. And in some species of orchids, you can also say that it is showing a polystylic condition. Now when we say about the pith may be present or it may be absent. If it is present, you can see in an example like maize, and if it is absent, you can say that it is example for that. If the pith is absent in a monocot root, then that will be EAMCET. The pith is sclerenchymatous in the roots of Typha and Triticum and Horridium vulgare. Here next one is the center is occupied by large number of metaxylem vessels there. For example, if we take an example of EAMCET, the xylem is hexarch with 2 metaxylem elements in the center. In this case, if the xylem is a hexarch and 2 metaxylem elements are in the center, then the pith is absent in this case. For example, in another example, if we take it is in grasses, the xylem is polyarch, and if it is a polyarch condition, then you can see the presence of distinct pith will be present. Okay, here there are 1 to 3 additional metaxylem elements will be present in the center, or if it is in the case of Avinia sativa. Okay, this is about your total TS of section of monocot root. Here we have studied about root hairs, which is absorptive organ, epidermis, which is a protective layer. Below the epidermis, some thick walled cells are present, those are called as exodermal cells. And again we have cortex. Cortex is round polygonal cells with lot of intercellular spaces are present in between them. Next is endodermis. Endodermis is uniseriate, it is common layer in all the roots, and it is consisting of universal occurrence. Next is endodermis, next pericycle, next vascular tissue system where we have xylem and phloem. Here the condition is polyarch condition. Next we have pith. Pith is sclerenchymatous, it may be present or it may not be present in some monocot roots. Okay, this is about TS of monocot root.